Okay, good evening, dear students. In last class, we discussed the estimation of iron or gravimetric estimations. Gravimetric estimations. First example, that is the estimation of iron from ferrous ammonium sulfate. The iron is present. How much iron is present in ferrous ammonium sulfate solution? In today's class, another example for common determination, that is second determination, that is a gravimetric estimation. In today's class, I am going to discuss about the estimation of sulfate from ammonium sulfate solution. Estimation of sulfate from ammonium sulfate solution using barium chloride. Here, the unit is gravimetric analysis. Procedure is same here, only chemicals is different. Using chemicals are different. For in last class we discussed the two lectures discussed the gravimetric analysis, that is iron. Here same principle, title, aim, principle, chemicals required and apparatus and procedure and observation, reaction, conversion factor, calculation and result. Here also same, and iron is also same. This is a barium sulfate, finding difference of sulfate. Here this name is gravimetric analysis. This is a gravimetric analysis here. The estimation of sulfate, estimation of sulfate, estimation of sulfate from ammonium sulfate solution, from ammonium sulfate solution, from ammonium sulfate solution using barium chloride, using barium chloride, BaCl2, barium chloride, BaCl2. Okay, this is a experiment, common determination of barium sulfate. Here using barium chloride, ammonium sulfate and barium chloride combined, the formation is barium sulfate. Here this is a title for this, and aim here we estimate the amount of sulfate. Aim is estimate the amount of sulfate gravimetrically. Estimate the amount of sulfate gravimetrically. Gravimetrically means in terms of gram. You weigh your substances, that is any of the residue. They estimate the amount of sulfate gravimetrically as barium sulfate, given solution, barium sulfate from given solution of ammonium sulfate, given solution of ammonium sulfate, this is a E. Here. Estimation of sulfate, you are estimating the sulfate in iron, you are estimating iron from given solution of ferrous ammonium sulfate. Here estimate the amount of sulfate gravimetrically. The iron is also gravimetrically, and here as barium sulfate from given solution you are finding barium sulfate, that is a sulfate from given solution of ammonium sulfate. Here you are giving the unknown is a ammonium unknown value. The ammonium sulfate solution is given into 50 ml volumetric flask. This is the aim and principle here. Principle is sulfate is precipitated. Here the sulfate is precipitated as barium sulfate. Sulfate is precipitated as barium sulfate by adding hot barium chloride. Here barium sulfate, the sulfate is precipitated as barium sulfate, adding the precipitated reagent, that is adding hot barium chloride. You are adding the hot barium chloride BaCl2. You are adding this BaCl2 and the sulfate is precipitated as barium sulfate and added this barium chloride solution to ammonium sulfate. Here this is added in ammonium sulfate. You added the ammonium sulfate. In ammonium sulfate, the barium chloride is added and the formation is here. The sulfate is precipitated as barium sulfate. In the pressure here, the presence of concentrated HCl. Here the presence of concentrated HCl of the solution and is after the precipitated barium sulfate. After this is filtered, residue is filtered, washed and dried, filtered, washed and dried, and afterwards ignited and cooled, and afterwards lastly weighed as a barium sulfate. This is you are weighing barium sulfate. Weighed as a barium sulfate. In iron you are weighing the ferric oxide. Here barium sulfate, in iron presence of ferric oxide. This is a principle. Next, chemicals required for this gravimetric analysis. Chemicals required here. Chemicals required: ammonium sulfate solution, ammonium sulfate solution, given. Ammonium sulfate solution given and barium chloride solution, 5% and concentrated HCl These are the chemicals required for doing this estimation, gravimetric estimations. That is the finding ammonium sulfate is given solution precipitated as barium sulfate. Next, apparatus. Same here, apparatus is 500 ml beaker with policeman, glass rod with policeman here, 500 ml beaker, glass rod with policeman, and watch glass funnel, asbestos sheet, watch glass funnel, asbestos sheet, watch glass funnel, asbestos sheet, and silica crucible, and desiccator, etc. And desiccator. Next is completed, principle is completed. Next, this is a chemical requires to be completed, apparatus is completed, finally process. You know the gravimetric procedure. Only chemicals are changes. The method is same. Chemicals are changes. Here, the ammonium sulfate solution. In that phase, ferrous ammonium sulfate solution. Here, barium chloride solution and concentrated HCl. The apparatus is also same. In this, the procedure, the dilute your given solution. Your unknown solution is given, that is ammonium sulfate solution. Ammonium sulfate solution is the unknown solution. The given solution is dilute. Here the unknown solution is given in a 250 ml standard volumetric flask. Right. Here procedure. First of all, you dilute the given unknown solution, the unknown ml of solution in a standard volumetric flask. You dilute that given solution up to the mark with using the distilled water. Now, given 250 ml standard volumetric flask, you dilute it with the mark. Here, this is a volumetric flask. Okay, this is a volumetric flask, and here the stopper is present here. You have here on volumetric flask the presence of this like a mark here. This is a mark. You add the distilled water up to here, up to this mark. Not one drop of upper. You don't miss one drop of lower insert. You add water on this mark exact for this mark. Up to dilute up to this mark with the distilled water. Dilute with distilled water. Distilled water. This is a 250 ml. 250 ml volumetric flask. Volumetric flask. This is a 250 ml volumetric flask. And then make it homogeneous. It is a shake well. Afterwards, you are diluted, added this distilled water up to this mass. After this mass, and after you shake this volumetric flask, you make this solution is homogeneous. You make it a homogeneous solution. It is a homogeneous. Afterwards, you pipette out this solution, this diluted solution. 25 ml of diluted ammonium sulfate solution. You take this diluted 25 ml. In iron, you taken 50 ml. Here also you take 25 ml. Here, this diluted solution is pipetted out 25 ml of this diluted solution, that is a ammonium sulfate solution. In a, you are already taken the clean 500 ml beaker. This is a 500 ml beaker. This is a 500 ml beaker. Here we add 25. We put out 25 ml this diluted solution and added here the 25 ml of this diluted solution in 500 ml beaker. And watch here with the glass rod, provided with the glass rod with the policeman, and watch plus cover with the watch glass with mouth, and here add 2 ml of concentrated HCl. Afterwards add 2 ml of concentrated HCl. Add 2 ml of concentrated HCl and 10 ml of distilled water. Here plus 10 ml of distilled water. 10 ml of distilled water. The same here in this beaker, 25 ml is diluted solution and 2 ml of concentrated HCl plus 10 ml of distilled water, and then heat the solution gently to boil. You heat this solution, presence of 25 ml this solution, 2 ml HCl and 10 ml distilled water. After completion of these added chemicals, afterwards you heat this solution gently for boiling, nearly to boil, and the solution here, the hot solution, you add 5% hot solution of barium chloride. Afterwards this is completed. Afterwards heating is completed. After this is heating to boil, heat to boil, and after this add this hot solution of barium chloride, BaCl, that is a 5% barium chloride. You have 5% barium chloride by drop in this 500 ml beaker. Slowly you add drop by drop barium chloride solution, and with constant stirring, you stir also with simultaneously, you add drop by drop and simultaneously you stir this solution. This is a glass rod. Here the presence of policeman here. This is a glass rod and used for stirring purpose. This glass rod with policeman and constant stirring till the precipitation of sulfate as a barium sulfate is obtained. Here the precipitate is barium sulfate, BaSO4, that is barium sulfate is obtained. And after the precipitation of sulfate as the barium sulfate is completed, and then add about 2 ml of barium chloride. Here afterwards add 2 ml of BaCl for excess. 2 ml of barium chloride, and afterwards added the excess of barium chloride, and then this beaker allow to the settle down here. Allow this for 10 minutes. Allow the precipitate to settle down. Test the supernet solution, and here the 10 to 15 minutes allow the settle down the precipitate, that is the barium sulfate is precipitate is settled down, and next we will check the supernet liquid. Here supernet liquid, above liquid, that is a supernet is that is a above liquid by adding the 2 to 3 drops of or 1 or 2 drops of this barium chloride eject the supernatant. The precipitation is completed or not. The completion of the precipitate, you add 2 to 3 drops. When in supernatant liquid, the precipitate is formed, then you repeat the experiment here. And then heat, then add barium chloride, then it is completed the precipitation. And here the barium chloride is added afterwards, the drop by drop for checking for precipitation. Then it is whether the precipitation is completed or not. If not, add few drops of hot barium chloride solution for the completion of precipitation. And digest the afterwards the precipitation process is completed. Then digest this precipitate or residue on a sandbar. After precipitation is completed, you check the supernatant liquid. After this process, you digest supernatant liquid on sandbar. Digest the supernatant liquid on sandbar for about 25 to 30 minutes. 25 to 30 minutes. 25 to 30 minutes. You digest this precipitate or residue of barium sulfate after this supernatant liquid and digest on sandbar for 25 to 30 minutes by taking care that the solution, that is, it does not boil to get a clear supernatant solution. Here it does not boil, it get a clear supernatant liquid and it settles this barium sulfate residue or barium sulfate precipitate. It does not boil, you take care for digestion purpose here. And then to make the residue is granular. Here it forms a takes place like granulars, and then afterwards you filter the supernatant liquid. Here the presence, here your precipitate is settled here, that is the barium sulfate, and here the supernatant liquid is present. Here the supernatant liquid is present. You filter this supernatant liquid or supernatant solution through an assless Wattmann filter paper number 40. Here we use Wattmann filter paper number 40. The number is 40. And wash the residue 4 to 5 times with hot distilled water. The residue is washed with hot distilled water. Hot distilled water. Wash with residue. Wash with residue. Wash with hot distilled water. You are using for washing purpose hot distilled water, that is you wash with 4 to 5 times with hot distilled water about 100 ml. Use about 100 ml. 100 ml of hot distilled water use, that is 4 to 5 times. 4 to 5 times. 4 to 5 times, and here until the last washings are free from chlorine by testing here. Okay. The last washing, up to you wash, the iron is here, the precipitate is free from iron, that is a chloride. Here it is tested with silver nitrate solution. Here free from chloride. free from chloride. This Cl chlorine here, chlorine molecule free from chloride, and this chloride test, that is you use AgNO3 solution. You are using silver nitrate 1 or 2 drops, and after the completion of washings, here the washings are completion, dry the residue. Here when the chloride is present, the ion of chloride is present. When chloride is present, then you repeat the washings until free from this chloride ions. Afterwards, this AgNO3 test, the last washings without precipitate, when no precipitate is formed, and then it completed, you confirm the completed the washings. Washing process is completed. And then washings are completed, dry the residue. You transferred your residue in conical flask here, in funnel. The funnel contain your filter paper. Here your washing is only 1/3. 1/3. Here this is a Wattmann filter paper number 14, and take this funnel. You dry this Wattmann filter paper. You kept this funnel on hot air cone. This is a hot air cone. This is a hot air cone. You kept this funnel on this hot air cone. Here this is the presence of, this is attached this funnel, and here the residue is present, and here when your residue is dried, it gets the cracks, and residue is dried, it gets the cracks. Cracks are formed on the filter paper, on Wattmann filter paper, and afterwards the drying is completed. Then the residue is along with filter paper. You transfer along with filter paper. You transfer with the crucible. That is the previously heated, cooled, weighed crucible. That is a silica crucible. Used as a silica crucible. Here we are using silica crucible. Silica crucible. Use this silica crucible and gently heat or ignite. Ignition means the red hot. Here ignite, and then in a electrical crucible heater. You take a electrical crucible heater, then heat this silica crucible. The presence here, the presence of this Wattmann filter paper along with residue. Here presence of residue, and this silica crucible ignition is completed. Afterward, the ignition is ignite 25 to 30 minutes. Ignite 25 to 30 minutes. Then after you remove your crucible on crucible heater, on electric crucible heater, then remove that crucible and cool the crucible extremely, and then in a desiccator. Here you cool this silica crucible in a desiccator for a room temperature. Afterwards, weigh the residue as barium sulfate, and here record the weight of the residue as a W grams. Okay, here weight is a W grams, and heating is a 25 to 30 minutes. After ignition is completed, and then remove the crucible. You transfer or you kept in a desiccator for cooling purpose when it cooled in a room temperature, afterwards you weigh the residue and it reported as a W grams of residue is formed here. This is a procedure for the estimation of sulfate from ammonium sulfate using barium chloride as a solution. This is a procedure for gravimetric analysis. Here I tell you once again, this is a procedure. Here first you take the unknown given solution, and you dilute that unknown solution. Afterwards the shake well because of the solution is make it a homogeneous solution, and the diluted solution is take 25 ml of this diluted solution in a 500 ml beaker. Afterwards you add 2 ml of HCl, concentrated HCl plus 10 ml of distilled water. After you heat till nearly boiling, then you add barium chloride 5% solution, and then the precipitate barium sulfate precipitate is formed, and after the barium sulfate precipitate is formation, then the adding this afterwards added this BSCl to barium chloride sulfate solution. Here the formation of barium chloride, and you add two test tube, nearly 2 test tube, and then add 2 ml of excess of barium chloride solution, that is 5% barium chloride solution. Afterwards this supernatant you kept at this heat this solution for you check for after for allow to settle down, then check the supernatant for completion of the this precipitation for 1 to 2 drops of the barium chloride solution for here you check supernatant liquid when completion of precipitation. Afterwards the precipitation process is completed, then you digest on sandbar for settled here for granules are formed because of this digesting this residue for the formation of granules here, and digest on sandbar for 25 to 30 minutes. Carefully to digest this precipitate because here nearly granules are formed for barium sulfate residue here. The granules are formed, then this precipitate is then allowed to settle down this precipitate for 10 to 15 minutes or 10 minutes. After this digestion is completed, for clear solution, you do not boil this solution. This solution do not boil. You digest carefully on a low flame, very low flame, because of air formation is there is a regular crystals of form here, regular formation, and then the here it get a clear supernatant liquid, and your precipitate is clearly settled in down surface of the here this beaker, and then this clearly settled, and you get a clear solution of a supernatant liquid after digestion. Then it get a supernatant clearly supernatant liquid, and this supernatant liquid is filtered. Then filtration takes place here. You take a Wartman ashless Wartman filter paper number 14. You filter the supernet liquid first, and afterwards the filtration is completed until you filter the ion is free. The chloride ion is free from last washings. You check last washings. You take a 2 to 3 drops of last washings. Use for washing purpose. You use the hot distilled water. Using the washing purpose hot distilled water. You wash 4 to 5 times for 100 ml. You use approximately or maximum 100 ml of hot distilled water for washing of precipitate or a residue here. Then the precipitated wash until this free from the chloride ions. When you check the last washing with silver nitrate, when you check the last washing with silver nitrate, it get a white precipitate. Then repeat this washing, and then washing is continue, and when the white precipitate is not formed, when you stop the washing, then you take a filter paper funnel, and then you dry the filter paper along here. Your residue is dry along with filter paper. You are on a hot air. This is a hot air form. This is known as a hot air. Here, this is funnel is present, and here you get the wire hose. Okay. Here this is a hot air core, and here the residue is dried along with hot air core. When residue is dried, it gets on filter paper, it gets the cracks. Then this filter paper this filter paper are transferred along with this filter residue is transferred along with filter paper on a crucible. Previously weighed then heated, cooled, weighed silica crucible. You are using in gravimetric analysis. This silica crucible, you transfer the filter paper and you fold that filter paper and transfer it in the silica crucible. Then silica crucible is heat to a electrical crucible heater. In electrical crucible heater, then ignite or heat the crucible, silica crucible, or ignite means heating takes with a red hot. If it gently near it is a red hot, and the filter paper is burns, then it deposited on the surface of the crucible is a carbon because of you are heating red hot. And then finally the crucible is, after ignition is completed, the silica crucible is removed. Then silica crucible is removed on electrical crucible heater. Then the crucible is transferred, or it is a silica crucible is kept in a desiccator for cooling purpose. Then it cooled in a room temperature, and then weighed your residue, and it reported as a W gram of the residue, that is the barium sulfate. Okay, this is a complete process. Okay, I will stop here only. In next class I will continue this process. This procedure is completed. In next class, I will take the conversion factor of this experiment, conversion factor, observation, reaction, conversion factor, calculation, and result. Stop here.